All right, everybody, welcome to part 5 of our discussion of all things neuro. This time we're talking about drugs. How can we interfere with the sort of normal neural processes? Now that we've talked about how neurons work, what are some ways that we have of altering that process of neural communication? So it's not just neurotransmitters that can affect receptor sites. There exist molecules that are able to bind to receptor types with some selectivity, and we call these ligands. So certain types of drugs can basically do what your regular neurotransmitters do. They can act just like them and bind to those receptor sites and produce similar effects. Alternatively, they can bind to the same sites and produce no effect or interfere with the functioning of that receptor. That's just one of many ways that drugs can work. Drugs work through many different mechanisms, and we're going to talk very briefly about some of the ways in which they can function. So, how do drugs do their thing? First, let's talk about general categories. Antagonists. These are drugs that block or inhibit neurotransmitter effects. So they sort of oppose the general effect of your endogenous ligand, your own neurotransmitters. Then there are agonists, which facilitate neurotransmitter effects. They might mimic the effect of the neurotransmitter themselves or make your own neurotransmitters more effective. So drugs can work in a lot of different ways. They can influence the production of neurotransmitters, right? So neurotransmitters are made within your neurons. So drugs can increase or decrease the amount of neurotransmitter that is produced. Storage and release, right? Neurotransmitters have to be packaged into those vesicles and then released to have their effect. Drugs can prevent that process or they can facilitate that process. They can work on receptors. In fact, a lot of drugs work at the receptor site. They can do a number of things here, right? They can bind to the receptor and mimic the effect of that neurotransmitter. They can just sort of get in the way and not do anything and sort of block your neurotransmitter from binding and doing its thing, or it can increase or decrease the efficacy of neurotransmitters at that site. Don't worry too much about all that. Just know that one of the sites that drugs can work is likewise reuptake or destruction of the neurotransmitter. I mentioned the example of a selective serotonin reuptake inhibitor earlier, which prevents the reuptake of serotonin. There are a number of different drugs that work through this mechanism, either facilitating or preventing the halting of that signal. So just to sort of hit this a different way, what I'm trying to emphasize is that drugs can impact the entire life cycle of a neurotransmitter. It just depends what sort of drug we're talking about. So normally, neurotransmitter is synthesized in the cell body or in the terminal, depending what neurotransmitter we're talking about. It's then packaged into vesicles. The neurotransmitter is released, and then when the vesicles fuse, then it binds and does its thing. Then it diffuses away or is metabolized or taken back up into the releasing terminal. That's normally the cycle of a neurotransmitter. I really don't want to go into too much detail about all this, but basically just know that at each of these stages, an agonist will promote this, right? So, for example, an agonist might cause more vesicles to bind and release neurotransmitter, right? Whereas an antagonist might prevent the packaging of neurotransmitter into a vesicle. Likewise, when it comes to binding, an agonist might mimic the effect of a neurotransmitter, or it might cause an increased effect at the receptor, whereas an antagonist might block the neurotransmitter from binding to the receptor at all. You don't have to memorize that specific stuff or worry really about the details here. I just sort of want to get the idea across that drugs can work at most stages of the life of the neurotransmitter. So here I like this figure. I just want to sort of use it to illustrate the idea of agonism and antagonism at the receptor level. So if you think about the agonist being the lock to the sort of receptor key, it sort of is the same shape and does the same thing as the normal neurotransmitter would do, right? This agonist is just like a synthetic key for this lock. So it binds, it's a perfect shape, and it produces the drug effect or the action, whereas an antagonist might work in the way of binding and that it occupies this site but produces no effect, right? In this way, your normal neurotransmitters can't get in here and bind because there's this thing in the way, right? That would be a way of antagonizing the receptor. So let's talk about a few examples of receptor interaction. So morphine is an agonist of the mu-opioid receptor. So at the actual mu-opioid receptor, morphine comes in and mimics the effect of endorphin. So it binds and acts just like endorphin and produces pain relief and euphoria, making it a useful analgesic drug. On the other hand, we have the antagonist curare. This is something that's found in a number of different plants, and certain indigenous populations actually use curare as a paralytic poison for hunting purposes, right? So it'll cause paralysis. This binds at acetylcholine receptor sites, but it has no effect. So it blocks the effect of endogenous acetylcholine. So one thing acetylcholine does, we mentioned very briefly earlier, is it's important for muscle contraction, right? So you need acetylcholine to your neuromuscular junctions to produce contraction of your muscles. Curare gets in there and prevents that from happening. So if you have curare in your system at your neuromuscular junctions, it's going to cause paralysis. You can't flex your muscles. This can be, of course, very interestingly enough, something that could be a cure is something like physostigmine, which actually reduces the degradation of acetylcholine. So normally, after acetylcholine signals, an enzyme called acetylcholine esterase comes along and takes it apart. What physostigmine does is prevents acetylcholine esterase from doing its thing. So basically, that means you've got more acetylcholine available in your synapses at your neuromuscular junction, which means that acetylcholine can outcompete curare and then allow you to move. All right, that's it for our brief discussion of drugs. Next time we are getting into the structure of the nervous and endocrine system.